is to work alongside individuals to serve their philanthropic goals while investing in VAI's life-changing research and education. Her role also includes connecting the community to VAI's researchers and educators through presentations like today. Kate brings over 25 years of experience in philanthropy, including serving as the campaign director at Hope College from 2002 to 2012. So welcome back. Kate will share a brief overview of the VAI, and then we'll hear from Dr. Henderson. Dr. Henderson is a neuroscientist whose focus is on the impact of protein pathologies and genetic risk factors on the development and progression of neurodegenerative diseases. He earned his BS in biological science from Florida State and his PhD in neuroscience from Yale. He then moved to the University of Pennsylvania for his postdoc fellowship. And in 2020, Dr. Henderson joined the VAI's Institute, the Van Andel Institute's Department of Neurodegenerative Disease Science as an assistant professor. Dr. Henderson's lab focuses on Parkinson's disease and Alzheimer's disease. Dr. Henderson will discuss how Parkinson's disease is triggered, how it progresses, and how that knowledge can be leveraged to develop disease-modifying therapies. So I'm going to turn it over now to Kate.
Okay, thank you so much for having us today. Like Tom said, I was at Hope for many years, so it's really fun to be back. And there are so many connections between Hope College and Van Andel Institute. We have many undergraduates that come and do summer research in our labs. And there's numerous things that we do in collaboration. So it's really fun to be here today. And so thank you for this opportunity. I just have a few slides before I turn it over to one of the brightest minds in Parkinson's research. So I just wanted to give an overview of what the Van Andel Institute is all about. We were established in 1996 by the Van Andel family. And so the Van Andels had a dream to create a medical research institute in our West Michigan community, but also to focus on science education. So we're pretty young. We just celebrated our 25th anniversary. And in that time, there's been some remarkable things that have happened. We've continued to grow. We have nearly 500 scientists, educators, and staff in the Institute, and they represent over 40 different countries. We've just recruited 6 new researchers and Michael was one of those, from really top-notch institutes around the country. So that's going to further our research that much more, and also our West Michigan community. We like to say that we're an institute with and without walls, because much of our work extends beyond the West Michigan community and the research that we do within the institute because of our global collaborations. Michael may touch on some of those today, but that really extends our reach because we're able to do things with global partners around the world. A couple things that I thought this group might be interested in is we have what we call our public lecture series, and those are free lectures multiple times a year, and they bring our researchers out into the community. We used to host those at the Institute, but now we are doing these virtually. So you can log in and listen for an hour. They're always at noon. And so you'll see there's several coming up. The next one will be May 19th. I won't read all these. You can go to our website and also see information about these and register, but they're going to cover anything from mostly cancer is the ones that will be coming up, but also our K through 12 education. And most of these researchers are fairly new to the Institute as well. And then I also wanted to share, because this group is focusing on Parkinson's research today, and Michael might talk about this as well, but on September 28th, we have our Grand Challenges in Parkinson's disease, where we bring together hundreds of scientists from around the world to look at the latest treatments in Parkinson's. There's also a component for people that are either living with Parkinson's or taking care of someone with Parkinson's called Railing to the Challenge, and that's also on our website. So if you're interested in that, it's going to be held at the Institute and you'd be welcome to attend. And then the final slide is just, again, to say thank you for having us today. We're really excited to be here. And this slide shares some different ways that you can stay in touch with the Van Andel Institute. You can sign up for our e-news. We have a blog, which has a lot of scientific information. You can follow us on Facebook or Twitter. And then if you have any questions following today's presentation or at any time, I put my email up there. Again, if you go to our website, you can find us, but you're welcome to reach out and we'd be happy to help you. So thanks again. I'm excited to hear Dr. Henderson's presentation.
All right, well, thank you so much. I'm happy to be here. This is my first visit actually to Holland because we moved here in August 2020. We've been to the down to Lake Michigan a lot. I'm really excited now to come here and talk to you all. Thank you for the invitation, Tom. So what I'll do today is I'm gonna go over some of the latest research in Parkinson's disease. I'll also touch on Alzheimer's disease as well. And I'm here for you. So if you have questions, please ask them and feel free to raise your hand if you have a question while I'm talking, if you want to clarify something. It's really, I left plenty of time for questions, so don't feel shy. So this is my lab. I like to start out by introducing them. These are the people that are in the lab every day doing the hard work. I've been really lucky since I came to the Institute to be able to recruit a really great group of individuals who are working every day to understand neurodegenerative diseases. And one of the ways that we do that is we try to understand how disease begins, how it progresses over time, and then we evaluate treatments for these devastating diseases. My lab is what's called a basic science research. So we do all the preclinical work to try and understand in tissue or in animal or cell models what's going on in disease processes. And then this is what most of the researchers at Van Andel Institute do. And then we can work later with clinical partners, so neurologists, to try and bring these things to the clinic. So things that I'll talk about today are what are neurodegenerative diseases, what causes disease, what are researchers doing, and I'll highlight some of the work being done at the Institute, but this is fairly representative of what researchers across the world are doing. And then I think one of the things I often get asked, and so I'll go into it into some detail, is how close are we to treatments that will slow or stop the progression of disease? So first, we're going to start off with what are neurodegenerative diseases? Well, in order to understand that, I first want to describe what the function of the brain is. So the brain is a wonderful network of billions of neurons. Neurons are the cells within the brain that do the functional work to bring us thought or movement. And what's important about the brain that's different than other organs within the body is that each region of the brain, it has a different function. So, for example, if you have in your liver, you have much of the liver has redundant function, so you can lose a part of the liver and you'll still be fine. The rest of the liver can pick up the load. It's not the same for the brain. So in the brain, each of these different regions, they may all look a bit like squiggly worms, but they each have an important and distinct function. For example, this one gyrus here is really critical for movement. So if you damage this region, you have difficulty moving. It's actually organized in such a way that this region down here controls facial movements. And as you move further down, you see trunk, these control your movement of your the body trunk. And as you go over the edge, that would control your legs. So this is each part of the region brain is very distinct. Right behind that is the somatosensory cortex. So this is where all the sensations, so touch and feeling and where your body is in space comes in and is organized. You all may know this is the frontal cortex, and the frontal cortex is really important for controlling the coordination of all these things and more complex. So you get sensations in from the environment, you then know how to respond to them. It's important for learning. So if you learn to play the piano, you have this premotor cortex, which really does a lot of the planning. So you can plan ahead which keys to play. And then in the very back of the brain, of course, because your eyes are up here, it makes sense. The very back of the brain is where you your visual cortex is. So the vision has to come all the way from here to the very back of the brain where it's sensed. This is sometimes when you can fall, you can see stars. That's because you've aberrantly activated that visual cortex. So these are all the parts that control higher cognition. Down here, this smaller part that's hidden is actually the part that controls really essential functions for our body. So breathing, your heart beating, sleep-wake cycles. These are all controlled by these nuclei down here, which is why if you get damage to this part of your brain, it's really deadly, because if you can't control your heart beating, it's essential for life. So, but what is even more important than the fact that each brain region has a different function is the fact that these all have to communicate with each other. You have to be able to sense that you've stepped on a tack and then respond to that by lifting your leg up. So the sensory and motor regions have to coordinate. And the way that our brain does this is that there are connections between each of these brain regions so they can communicate. It's much like a telephone pole to be able to communicate from one region to the other. Now we all rely on cell phones, but unfortunately our brain doesn't have wireless communication. All the communication in the brain has to be wired. This brain region has to coordinate with this brain region. And so you can see it sends a long track of wires or axons from one brain region to the other. So that's the brain. And here you can see the rest of the central nervous system. This is how the brain communicates with the rest of our body. And in neurodegenerative disease, the brain or the central nervous system can dysfunction, and that can lead to motor or movement dysfunction or cognitive dysfunction. And two of the most common of these neurodegenerative diseases are Alzheimer's disease and Parkinson's disease. Alzheimer's disease is a form of dementia that interferes with memory, thinking, and behavior, while Parkinson's disease can exhibit movement problems such as tremors, difficulty balancing, and difficulty walking. Interestingly, for Parkinson's disease, we think of this as a movement disorder, but it can also exhibit several other symptomatic dysfunction, including gastrointestinal dysfunction, loss of sense of smell. And as a part of disease progression, many of Parkinson's disease patients can eventually in end-stage disease go on to develop dementia as well. So I told you I'm a basic clinical researcher, so I wanted to give you, let you hear a little bit more from a movement disorder specialist. Let me see if I can click off of my, there we go. Okay, so this is from Rachel Dalhoun. It's through the Michael J. Fox Foundation. If you don't know about Michael J. Fox Foundation, this is an organization which supports Parkinson's disease research. They have a lot of great educational materials. I thought this was a really excellent video, so I'll just play it for you now.
Hi, I'm Dr. Rachel Dolan, Vice President of Medical Communications at the Michael J. Fox Foundation for Parkinson's Research. I'm also a movement disorder specialist, which means I'm a neurologist with specialized training in Parkinson's disease, or PD. Today, I'd like to discuss a question that is essential to our foundation's work. What is Parkinson's disease? Let's start with some basics. In the United States, it is estimated that anywhere from 600,000 to 1 million people or more are living with Parkinson's, and at least 60,000 are diagnosed each year. This makes PD one of the most common brain diseases, second only to Alzheimer's. Age increases the risk of Parkinson's, and the average age at diagnosis is 60. So as our population continues to grow older, more people are likely to experience PD. In fact, people tend to think of Parkinson's as an older person's disease, but some get PD at 40 or even younger. Some diseases can be diagnosed with a lab test. Cholesterol levels and blood pressure are measured to evaluate for heart disease, for example. We need tests like that in Parkinson's, but they don't exist yet. Doctors diagnose Parkinson's by completing a medical history and a physical examination. They look for 2 of the 3 classic motor symptoms, which are resting tremor, stiffness, and slowness of movement. When people hear about Parkinson's, they mostly think of these motor symptoms, especially tremor. But some people also experience walking and balance problems, and PD affects other body systems. Constipation, sleep problems, cognitive changes, and depression can occur, sometimes even before a Parkinson's diagnosis. And some people report that they lose their sense of smell. One of the hardest things about Parkinson's is that everyone with the disease embarks on a unique journey. In fact, movement disorder specialists say, if you've met one person with Parkinson's, you've met one person with Parkinson's. Each person has their own mix of symptoms, and there is no standard trajectory or path. So if everyone gets their own version of Parkinson's, then what does everyone with PD have in common? To answer that, let's take a look inside the brain. In Parkinson's, the brain cells that make dopamine stop working or die. Dopamine is a signaling chemical or a neurotransmitter that coordinates movement as well as feelings of motivation and reward. When dopamine cells die, Parkinson's symptoms emerge. Exactly why these cells die is not well understood. Researchers believe that in most people, Parkinson's is likely caused by a combination of genetic and environmental factors. But although certain environmental factors, such as pesticides and head injury, are associated with an increased risk of PD, in most people, there is no clear exposure we can point to as a straightforward cause of their Parkinson's. Similarly, while genetic mutations are linked to an increased risk of Parkinson's, based on what we know today, the vast majority of cases are not directly related to genetics. However, the field of genetics is moving fast. In Parkinson's, as in many other diseases, tremendous research is focused on genetics because this is our best opportunity to uncover paths to treatment breakthroughs. But how do we treat Parkinson's today? Here's what I told my own patients when I was a doctor in clinical practice. The currently available PD medications can't slow or stop the progression of the disease, but they can ease symptoms and help you continue doing much of what you have always done. The same goes for certain surgical procedures, which are a good option for some people. The most important point is that treatment regimens need to be individualized, so you should work closely with your doctor to determine what medication or combination of therapies work best for you and your symptoms. Speaking of doctors, our foundation urges people with PD to see a movement disorder specialist if possible. Many people don't get to these experts, and we'd like to change that because the right doctor can help optimize your Parkinson's care, update you on research and drug development, and direct you to clinical trials, which are vital opportunities for you to advance scientific progress. Specialists can help you build the right care team and treatment regimen, but there's much more you can do to feel your best, like making sure to eat well, exercise, and stay connected to others. Check out our full Parkinson's 360 toolkit for advice on living in the here and now with Parkinson's, as well as information on what to expect as Parkinson's unfolds over time. So, what is Parkinson's disease? Today, it's different things to different people, but by working together urgently and with purpose, we hope to make it a thing of the past for everybody. At the Michael J. Fox Foundation, our mission is nothing short of eradicating Parkinson's altogether. Much work remains to be done, but we've got many reasons to be optimistic. As Michael J. Fox says, the latest research is the latest hope. We're closer than ever to new and better therapies, even ones that could slow or stop the progression of the disease. And until the day we find the cure and close our doors, we promise we won't stop.
Okay, so I thought that was just a great overview. Kind of gives you an idea of when this disease begins in many individuals, what kind of symptoms are exhibited, and treatments that are currently available. And so now I'll go on to a little bit more detail about what we think is going on in the brain. So what causes these diseases? So as Rachel had emphasized, we don't know everything about the disease yet, which is why we're doing research, but many years of research have begun to discover some of the underpinnings of disease. So as she had mentioned, there's this important part in the brain called the substantia nigra that houses a type of neuron called dopamine neurons. And that one consistent feature across Parkinson's disease is that these neurons are lost. So you can see in a healthy substantia nigra, these neurons are actually pigmented, so you can see them, they're dark within the brain. And then as Parkinson's disease occurs, many of these neurons are lost. And so she mentioned that one of the treatments that can be used is called levodopa, and it basically supplants the dopamine that's released by these neurons by directly giving this to patients. Another hallmark of disease is this accumulation within cells called Lewy bodies. You may have heard of these, and we'll talk a little bit more about what they are. But we think that these Lewy bodies are a result of a disease process that leads to loss of neurons within the substantia nigra. And importantly, as disease progresses, these can be found in many other regions of the brain. And we think that that's related to the dysfunction that's seen as Parkinson's disease progresses. So what causes disease? Within those Lewy bodies, there are certain proteins. And so we can understand more about disease by investigating these proteins. One of those is called alpha-synuclein. This is a protein within the brain that has important functions, but as a part of disease, it becomes misfolded. So you can see normally it has this really nice curly structure. And as a part of disease, it has this now kind of beta sheet, it's called, structure that is kind of crumpled and folded. And as time progresses, these misfolded proteins can accumulate into elongated fibrils. And so I like to analogize this misfolding of protein to paper. Paper is something that we all use in everyday life. We can write on it, we can print on it, we can paint on it. So it's very useful. We keep reams of this paper around our office or in our homes. So this is like proteins. Proteins have an important function in the brain. But what happens with these proteins is they become misfolded. And for paper, you know that this now makes the paper not useful for any of the previous purposes. And so we throw it in the trash bin. Well, the cells in our brain know to do the same thing. When that protein becomes misfolded, we throw it in the trash. The problem is that with age, our trash clearance system gets less good, and those proteins can accumulate, much like if the trash was not taken out at your house or in your office, and you would slowly accumulate these things, and they can lead to disruption of the cell's function. So in Alzheimer's disease, these toxic proteins are known as tau tangles, shown here on the left. Let's get back to the laser pointer, or amyloid beta plaques. You can see that tau tangles occur within these neuronal cells, and amyloid beta plaques are in the extracellular space. In Parkinson's disease, as I had mentioned, this is a picture of an actual alpha-synuclein Lewy body and a Lewy neurite from patient tissue. So one of the things we do in my lab is try to look in the brains of people that had passed away, in this case from Parkinson's disease dementia. We can see this is one of those cortical regions that's important for the function of individuals. And in green, you see many of these neurons. So when I told you there are many neurons, this is a tiny bit of the brain, and you can see how many of these are. There are billions within the brain. And so what's different between a healthy individual and a person that is exhibiting Parkinson's symptoms is this magenta color. So this magenta, these are Lewy bodies and Lewy neurites within this patient brain. And if we take away the image of those individual neurons, you can just see how chock full that brain has become with these toxic proteins. So the brain is no longer clearing those proteins from individual cells. And so it accumulates, and we think this leads to dysfunction and eventually neuronal loss. So again, as I had taken some time to emphasize that each part of the brain has a different function, and that dysfunction of any one of those regions can disrupt function in the other regions, because we know that for disease, where in the brain also matters. So if you have accumulation of these toxic proteins in a region that controls memory, there's memory dysfunction. If it's in a region that is controlling motor function, like those dopaminergic neurons I told you about, then you have motor dysfunction. And further, we know that over time, for reasons we don't quite know yet, those protein pathologies can progress to different regions of the brain, and you can exhibit symptoms associated with disruption of those different regions. So how does this process begin? Well, we don't know everything about disease. What we know is that some of it we're born with, it's in our DNA. There can be familial forms of Parkinson's disease, but these familial forms are quite rare, but you can still have certain genetic risk factors that predispose disease. And then the other, of course, is environmental risk. So we're all exposed to different chemicals or potentially different infectious disease that give us some enhanced risk. Importantly, as Rachel had mentioned, we don't know, none of these, for the most part, would cause disease that would enhance your risk. So instead of in the general population, everyone in this room would have a 1% chance of developing Parkinson's disease throughout our life. On average, But these risk factors could increase that. Maybe now you have a 2 or 3 or 4 or 5% chance of developing disease. So the title of the talk is the latest in Parkinson's disease research. What are researchers doing? And I'm going to highlight some of the research done at Van Andel Institute, just because this is some of the things I'm most familiar with. And I'd like to introduce you to some of the researchers at the Institute. But much of this is representative of what's being done across the field, nationally and internationally. So this is our Department of Neurodegenerative Science. It's led by Dr. Darren Moore, who's our chair. We have currently 8 members. Patrick has recently left. He's now an adjunct professor and still involved with the Institute. And I'll tell you a little bit about one of the things that he has done in his time here to help bring our research to clinical trials. But our research encompasses several different things, including genetics, environmental toxins, epigenetics, and I'll talk about that, what all of these are in a minute, cell biology, neural circuits, and we also do animal modeling. And while we don't do clinical trials at the Institute, we help facilitate them. So genetics, as I had mentioned, all of us are born with the genes we're going to live the rest of our life with. And for some of us, we have certain differences in those genes that can cause disease. But much more commonly, all of us have different genes. That's why we all look different, we sound different, and we're all from different families or different regions of the world. And so each of us has some different risk level just based on which genes we are born with. And so what labs like Jose's and Rita's do is they look in gigantic populations. So these are tens of thousands of people with Parkinson's disease or without Parkinson's disease, and try to understand what genetically makes those people different. Often that's very small changes. So it's something that would give a person now, instead of a 1% of chance of getting Parkinson's disease, maybe a 2% chance. And then labs like Jerry's take those genes and try to understand what those genes do and how they could possibly precipitate disease. I had mentioned environmental toxins, and Lena and I are part of a group called IPARC, which is infectious triggers of Parkinson's disease. There are known environmental toxins that can enhance risk of Parkinson's disease, but for most of these, we don't know how that occurs. So certain bacteria, virus, or chemicals can be associated with increased risk. And it's difficult to understand sometimes why these cause disease. So we can look in our different models to understand what changes when you expose cells or animals to these pathogens and that could possibly predispose to Parkinson's disease. This is a really interesting area of research because we have the ability to control often what environmental exposures we are exposed to, or to treat, for example, if there are certain known pathogens that could cause disease. So epigenetics is a form of genetic research. What it does is that it relies on which genes are expressed. And so epigenetics is what makes your heart, and your brain, despite the fact that they have the exact same genes. It controls which of those genes express and makes a cell turn into a heart cell or a cell turn into a brain cell. And so this is really important because I told you, while there is some genetic component, we think for Parkinson's disease, that it accounts for 25% or less of our risk of developing Parkinson's disease. And one way the environment can influence genes is through epigenetics. So this basically modifies DNA to make certain genes express or not express. And this is a great way to understand how environment could modify our genetic profile. So again, Rita and Jose are looking at what these modifications are, and Jerry's lab tries to understand how that can impact disease. So the fundamental building block of all biology is the cell. And so we can look at individual cells and processes going on with individual cells to understand which molecular pathways may be disrupted. For example, if we take a cell from an individual that had Parkinson's disease and compare it to a cell from an individual that did not, can we understand fundamental cellular processes that are different in those two cells? And this is a really great way too, because we can culture these in dishes with 384 wells or 96 wells. And so this allows us to screen a lot of potential therapeutic molecules. This gives us really high throughput and a rapid way to identify potential therapeutic molecules. But of course, cells don't exist by themselves. We're not single cellular organisms. We have trillions of cells in our body, and they all have to interact together, especially in the brain. I told you the intercoordination of those cells is really critical to the function of the brain. And so labs like Hongyuan and mine try to understand how the brain functions as a network of interconnected cells. And we do that to try and understand questions like how protein pathologies progress through the brain over time. And can we restore normal function by modulating those circuits? And in fact, previous research done in the Parkinson's disease field has shown that we can do that to some extent. So you may have heard of deep brain stimulation, and what this is essentially our effort to modulate the way the brain functions in a circuit level by enhancing electrical activity in certain regions. We also, after we find certain things in cell models, we often will bring them to animals because animals are much more complex and allows us to test some of our potential therapies in much more complex systems. We can understand, for example, how genetic factors and environmental factors interact within a single organism. And then we can also assess our most promising therapeutic molecules for their ability to reverse the effects of disease in our disease models. And then finally is clinical trials. So I mentioned the Van Andel Institute does not run clinical trials. We are a research site. And so, but we're really passionate about the ability to bring therapies to patients. And so Patrick has really spearheaded this through what is known as the Cure Parkinson's VAI International Linked Clinical Trials Initiative. So this is a partnership between Van Andel Institute and Cure Parkinson's in the United Kingdom. And what they do is they bring researchers from all over the world together to discuss molecules that have already been approved by the FDA for other disorders. So they're known to be safe and able to be used in patients, and now see if there's some evidence from research being done that these could be efficacious for Parkinson's disease. What this does is allows us to rapidly progress those molecules through clinical trials as potential new therapies. And I'll talk a little bit more in the session at the end of the session when we discuss how close we are to therapies for Parkinson's disease. So that's a summary of our department and many of the things we're researching. We've been able to bring together a really great group of individuals who are hitting all these main topics in Parkinson's disease research. We often work together. So as I had highlighted for each of these, there are multiple people discovering genetic risk factors, other people trying to understand what those do, and then many people then screening therapeutic compounds or trying to bring to understand if those would be efficacious in animal models. So another thing I wanted to highlight is a much more recent addition to Van Andel Institute's program, and this is through philanthropy from the Van Andels to develop the West Michigan Neurodegenerative Disease Program. And what this is really doing is the Van Andels have put a lot of effort into facilitating neurodegenerative disease research. They want to speed this up. We're all looking for the answer as quickly as possible. And so the mission is to provide critical resources for VAI scientists to simulate leading edge neurodegeneration research. And the main initiatives in this are that we're developing a biofluid biorepository, so that is collecting blood from individuals to understand if we can find biomarkers, for example. So Rachel, in that introduction to Parkinson's disease, had mentioned that we don't have biomarkers. So you can't go to the clinic and just get your blood taken and we can tell you that you have Parkinson's disease. Can't tell you how fast you will progress, but there's promise that possibly in the future we will be able to do those things. We also have a brain biorepository. So by studying the brain of individuals that had Parkinson's or Alzheimer's or other neurodegenerative diseases, we hope we can understand what went wrong in the hope that we can reverse that. And then finally is iPSC platform, and I'll talk about what that is, but it's essentially taking patient cells to understand at the cellular level what may be going wrong. I'll just go into these really briefly. We've just started this over the past year, but one of the things that we're doing with local partners is to collect biofluids from neurodegeneration to start a neurodegeneration specific biorepository and perform population-based sequencing. So I had mentioned that individuals can, by studying the genetics of many large groups of people with neurodegeneration, we can understand what may be predisposing them to disease. And then identify genetic variants, such as some of the ones I'll talk about soon, that can cause individuals to have increased risk of disease. Our brain biorepository, we've recently started. Its purpose is twofold. So one is that you can be diagnosed clinically, but you won't. We don't know what disease you had until we get a final postmortem neuropathological diagnosis. And the hope is that we can provide this for clinicians so they'll know, have a better idea of what disease patients had. And then also, as I had mentioned, help researchers uncover what happens in the brain during disease. iPSCs are induced pluripotent stem cells. These essentially, we can take some skin cells from a person or even blood cells, and then induce those into what's called a pluripotent state. So it means that those cells can differentiate into different cell types. So we can try to form, for example, neurons in a dish, but from an individual who had disease or an individual who didn't. And it allows us to, again, understand those fundamental cellular processes which may be disrupted in disease. So that I just wanted to highlight that recent initiative. And then I'm going to tell you a little bit about what my lab research is. These are the things that I'm most passionate about, and I hope that are making, we can make a great impact on the field. So we research several different questions. We try to understand how those pathological proteins can spread through the brain over time, how genetic risk factors may impact disease, which cellular pathways are disrupted, especially by these protein aggregates that accumulate in cells. And then we put an emphasis also on testing potential therapeutic molecules that could slow or stop progression of disease. So some of the things that we found recently are that protein pathologies spread through the brain using those same neuroanatomical connections or those same wires that are important for brain communication. So it seems like even if pathology begins in a region deep down here in the brainstem, it can then follow some of these connections up through the brain. And this, we think, leads to progression of disease. However, one of the interesting things we've also found is that not every region along those highways will develop disease. Some of them are resistant to developing it. And we think we can find clues from those regions that are resistant to perhaps make the whole brain resistant to developing pathology. We do this using a combination of what is called quantitative pathology, that is defining exactly how much of these misfolded proteins there are in every region of the brain, and through network modeling. So network modeling is essentially a method to think of the brain regions and the connections between those brains as a large network of interconnected hubs. So for example, if we have protein pathology that spreads up through here, we think that it has to go through certain hubs, and perhaps by treating certain hubs that are important for that progression process, those are the best places to intervene in treating disease. We also study those genetic risk factors. Two of the most common genetic risk factors for development of Parkinson's disease are called LRRK2, this is leucine-rich repeat kinase 2, and GBA. If you have one of these mutations, you have an increased chance of developing Parkinson's disease. And what we think that they're doing from our research is that they're reducing the ability of the cell to clear those pathological aggregates. They're reducing the frequency that the trash is taken out of the cell. They also may change the speed of the spread of pathology through the brain. So instead of very slowly progressing over perhaps 10 or 15 years, we think that some of these genetic risk factors actually speed up how quickly the pathology can progress and leads to a more rapid progression of disease. We also look really deeply into those aggregates. So we know that there's fibrils of these misfolded proteins, but it seems like other proteins are getting stuck there. They're mistakenly discarded as part of this trash. and they may lose their function. So perhaps by enhancing the function of those proteins, we may improve disease. So of course, once we know what's going wrong, we can develop targeted therapies for these pathways. We can try and reinforce those cellular pathways so those protein aggregates are cleared. We can reverse damage caused by genetic risk factors by targeting those genes that are being most impacted. And we can slow or stop the progression of spread. So in the final part of my talk, I wanted to answer the question that's most often asked, which is how close are we to treatments to slow or stop the progression of disease? So I like showing this figure. I know it's very complicated. I'm going to walk you through it. These are current clinical trials for Parkinson's disease. With each one of these different words is a different compound or treatment that's being investigated for Parkinson's disease. The way the clinical trials work is there are 3 primary phases. Phase 1 is simply a safety testing. So this is in a very small number of individuals who don't have disease, just to make sure that we can get a dose correct that doesn't show any adverse side effects. Phase 2, if a compound passes phase 1, it is safe, we can administer it to people. Then it goes to phase 2, which is further safety testing. We'll do this in a slightly larger number of people. And in this case, it may go on into patients. So we would test in patients, number 1, can they tolerate this type of treatment? And we can get some preliminary evidence whether it changes the disease course at all. And then phase 3 is the gold standard of clinical trials. This is done in double blind. So the patient doesn't know what compound they're getting. The doctor administering it doesn't know what it's getting. That's why it's double blind. And there's a comparison to placebo. So you would get either the compound that we think is therapeutic or some compound that has no target engagement. And these are large. They're done in hundreds of patients, often across multiple centers across the country, across to really assess, does this change disease progression? And so if you look at this diagram now, the outer ring is phase 1. So these are ones that are just in safety testing. The middle ring is phase 2. So this is further safety testing and perhaps some early evidence whether it's efficacious. The inner ring is this final and this phase 3 to see if compounds change disease trajectory. And the other thing you should note is that the bottom half of the circle is symptomatic therapies. The top half are disease modifying. So symptomatic therapies are things like levodopa that can alleviate those symptoms and give you a better quality of life, but it won't change the overall trajectory of disease. These disease modifying therapies are things that we really hope can actually slow or pause progression of disease so that individuals will live out a normal life. And so you can see, I think it's a hopeful time in Parkinson's disease research. There are more compounds, more promising therapies that are in these clinical trials than we've ever had before. Although you will see also that this phase 3 is quite sparse for disease modifying therapies. We currently have 2 that are in phase 3 clinical trials. It's very difficult to bring a compound through all these, and it can take many years. So it's a long time to bring a compound to clinic. And unfortunately, to this point, no compound has made it all the way through phase 3 for disease modifying therapy. So we don't have a disease modifying therapy yet. One other thing you'll note is that there's a color scheme here. So each of these different colors is a different target. So I told you one of the targets that we know has some genetic rationale is GBA. There are many, about 10% of Parkinson's disease patients will have GBA variant. And so we think these are potentially therapeutic even for people with idiopathic or non-genetic forms of disease. And so some of these you can see now are moving into phase 2 and phase 3, and I'll highlight one of these in a minute, ambroxol, which was advanced by the Link clinical trials, International Link clinical trials. Many other different compounds, some targeted at those pathological aggregates themselves through immunotherapy, some at kinases like LRRK2, I had mentioned, also has genetic support. And so one of the things that Van Andel Institute has done is to partner with Cure Parkinson's into these international linked clinical trials. As I told you, clinical trials, pushing a compound through from phase 1 all the way through the end of phase 3 is a very long process. And so to speed this up, scientists have advanced compounds they think may have an effect, but that have already been approved by through those safety trials. One of these is ambroxol. So this is a mucolytic that's used to treat bronchitis. What does this have to do with Parkinson's disease? Well, it turns out that ambroxol, while it's used to treat bronchitis, also happens to hit glucocerebrosidase, which is encoded by GBA1. So it seems like, especially in individuals that have a GBA mutation, that this ambroxol may help enhance function of the enzyme. And so it may be useful for treatment of Parkinson's disease. Several of these others, you can see some of these are diabetes drugs. Exenatide is one of the 2 compounds that's currently in phase 3 clinical trials. It was originally discovered and used as a diabetes drug, but it's shown some promise in preclinical models and even some preliminary evidence from clinical trials that it may be useful. And so that's why it's now in phase 3 clinical trials. So international linked clinical trials, again, is a way to try and speed up how quickly we can move some of these into treatments for Parkinson's disease. I also wanted to highlight Alzheimer's disease treatments. So these are several, you can see again, this is phase 1 on the outside, phase 2 on the middle, and phase 3 on the inside. This is broken down slightly differently, that we have symptom reducing small molecules and then disease modifying. You can see there are many more disease modifying treatments for Alzheimer's disease currently in the clinic. And so this is broken down into biologics. So biologic basically means that we have to make this in some way in a cell system, for example, instead of small molecules, which we can simply synthesize in the lab. And you can see that there are a large amount of these, including in phase 3 clinical trials. You may have heard of some of these. Aducanumab recently was preliminarily approved, and now it's decided that essentially it's going to go through what's dubbed a new phase 4 clinical trial, which is there's some evidence that it's efficacious. It definitely is hitting its targets, clearing some of those amyloid beta plaques from the brain. But there's also not as much evidence yet that it is modifying symptoms. And so the regulators have decided that it should be shown, have additional evidence shown that it's efficacious before it's used more broadly. But Alzheimer's disease, I will say, is a little bit ahead of Parkinson's disease research. That's why there's so many in phase 3 clinical trials, because we simply knew more about the disease earlier. And we're getting there with Parkinson's disease now through all the research that we're doing. And so I have hope that Parkinson's disease will also soon be here. And there's a lot of hope, especially in the Alzheimer's disease community, that we may very soon have an actual disease modifying treatment. Because for Alzheimer's disease, unfortunately, different than Parkinson's disease, they're not as great of symptomatic therapies. So we don't have as great therapies to kind of treat symptoms for such a long period as we do for Parkinson's disease. So I wanted to leave plenty of time for you all to ask questions. So I'll just go ahead and conclude. So in conclusion, I hope some of the things that you've gotten out of the talk today are that the brain is a wonderful network of billions of neurons that must communicate properly through these connections in the brain to form even the simplest thought or movement. When proteins within the brain, which are important for its normal function, misfold, they can perturb the cellular function within the brain, and whole circuits can be disrupted, leading to what we see symptomatically as a neurodegenerative disease. Scientists at Van Andel Institute and across the world are trying to discover how this starts and progresses with the goal of stopping it. And my lab is amongst those that are trying to uncover what happens in disease. And I'm happy to answer any questions you may have.
Thank you.
How long has Parkinson's been known as an entity?
Yes, so Parkinson's disease was first described in the 1800s by James Parkinson. That's where it got its name. But at that time, he described many of the symptoms, but we didn't really know what was happening. In fact, not until the past 30 or 40 years did we know that it was the dopamine neurons that were lost in the brain that were responsible for that. The person that discovered that, was the dopamine that was responsible. They actually went on to win the Nobel Prize for that discovery. And then at that time, when that was discovered, it seemed that possibly dopamine replacement therapy was a way to treat patients. And so that was also one of the other major discoveries in the field. But understanding more of the biology underlying what actually happens in Parkinson's disease is very recent. Only in the past 20 to 30 years have we really started to dig in and understand at the cellular level what happens in disease.
Is Parkinson's about equally divided between male and female?
Parkinson's disease is slightly more prominent among men, so it's biased in that direction. Alzheimer's disease is actually slightly the opposite, so it's slightly more common among females.
You've listed the array of symptoms that are used to diagnose the disease for Parkinson's. What normally brings patients in first?
That's a great question for a neurologist. So I don't typically see patients, but I think oftentimes it is one of these movement disorders. So what we know about disease, interestingly, is that many of these other symptoms can occur long before the movement symptoms. So for example, constipation can happen 10 to 15 years before onset of movement symptoms. Sleep disturbances also can occur 10 years or more before patients will actually have sufficient motor symptoms to come to the clinic. But it is typically my understanding that it's the motor symptoms that will bring patients to the clinic. And then it's at that time, it's seen that it may have been progressing actually for some time based on when these other symptoms occurred.
How about the slowness?
Yes, so slowness of movement, and as the neurologist had said in the little video that I had played, I think it's a little different for every individual. So some people may come in for a tremor, or they may come in for slowness of movement. They may come in because they have some gait, maybe they're falling due to gait disturbances. But I think it's different for every patient.
You mentioned that simvastatin, the statin medication, is being looked at for possible benefit for Parkinson's treatment. Does that suggest that there's an inflammatory dimension to Parkinson's?
Yeah, so for simvastatin, I'm not as familiar with that literature exactly why that's being pursued, but there is strong evidence that there is some inflammatory component to Parkinson's disease. So for sure, some of these, for example, infectious diseases that could lead to a systemic inflammation, that's possible that that's how they're predisposing to Parkinson's disease. If we look in the Parkinson's disease brain, we can also see, especially in those areas that have pathology, that there is an inflammatory response within the cell. The brain has its own resident kind of macrophages that respond to neural injury or things like that. And there is some response within the brain that would suggest there's an inflammatory process going on. And within preclinical models, there's some evidence that reducing that inflammation may be helpful. So it's possible, but I can't speak specifically to the background for that compound.
So we've got a question from Zoom. You mentioned movement disorder specialists and that not enough patients seek them out. How rare are these specialists, and do we have any in Southwest Michigan?
Great question. So this is, I think, as she had mentioned, especially in rural communities, it's very difficult to find a movement disorder specialist. I know that in Grand Rapids, for example, there's a very nice movement disorders clinic. I know some of the people that work there, but I think these are much more common in metropolitan areas, and it can be much more difficult to find this kind of clinic in more rural areas. But yes, there are these clinics in Southwest Michigan. So So within 45 minutes of here, for example.
You mentioned the biofluid biorepository, and you specifically mentioned blood.
Yes.
Is cerebrospinal fluid being looked at as well for biomarkers?
Yes, definitely. So, so as I had mentioned, Benin Institute, I don't know if we mentioned the exact size. There's around 500 people working there, but there's only around 35 labs. And so one of the things, so at big clinical centers, so for example, you know, at a big clinical center like where I was before, at the University of Pennsylvania, there's neurologists. We have big research projects that may be involved in collecting cerebrospinal fluid. For the MIND initiative right now, we're focusing on blood because that we don't have such a huge presence to kind of develop these clinical tools and clinical research tools that would collect cerebrospinal fluid. As you may know, it's not so simple as just going in and you get a little blood taken. The cerebrospinal fluid collection is much more involved. So you really need people dedicated to that kind of project. And right now we don't have that. It's certainly something of potential interest in the future. But right now for the VAI MIND initiative, we're focused on blood because it's a lot easier to collect that and less invasive.
I'm Suzanne Harrington, and I'm just wondering, I need a refresher course on what dopamine is in the system. What is it about?
Yeah, so dopamine is, in that video, Rachel very quickly said it's a neurotransmitter. And so what neurotransmitters do is they essentially allow neurons to communicate with each other. So throughout much of the brain, there's excitatory neurotransmitters, so neurotransmitter called glutamate. And this basically allows the neurons, so one neuron fires, tells the other neuron to also fire. So like a sequential firing. Dopamine is a little bit different in that dopamine can have different effects depending on the neuron that it's talking to. And so what essentially does is we have motor circuits which tell our body to make certain movements. But what the motor cortex does is it basically just says go or don't go. But what the dopamine does is it refines those movements. So it says go, but do it slower or faster. So we can do very smooth fluid movements is basically what dopamine's role is there. It kind of controls the tone of the system. So you can make very smooth movement. Yes. Yep. So your brain basically without dopamine can still tell you to move, but it's a problem sometimes of initiating that movement or the smoothness of that movement.
Any other question? I have a couple of questions, actually. I just need to get back and read them.
And I think we had one back there
Oh,
too,
okay.
as well,
Yep.
Tom.
My husband is involved in Evergreen, and they are having a lot of success there with the pedaling for Parkinson's. I'm wondering what your take is on exercise and how that is affecting the Parkinson's.
Yep. So there's a lot of evidence actually that exercise can help, which is a little counterintuitive. You would think for a movement disorder, if it's difficult to move, that it may be difficult to exercise. But you said pedaling for Parkinson's. One of the things that's interesting is that certain motor circuits, for example, cycling, even while it may be difficult to walk well, a lot of patients still find it easier to do certain learned motor kind of circuits, such as cycling. Some people even, I know one patient that still finds it very difficult to walk, but can play basketball well, can dribble the basketball, just because it's a learned motor thing. It's wired in a certain way within the brain that allows him to do that function, even while it's difficult to do kind of things that like volitional. So if you decide that you want to walk versus kind of doing something that's more automatic, it's much more easy. And that's a great way for patients to get exercise. Also, I know some dancing with Parkinson's. So definitely it's well known that exercise is beneficial.
I have a question. and I'm going to spell this word for you because it was on the board, EXENATIDE.
Yep.
And it was used for diabetes. Have you found in your brain bank and the brain banks of the diabetic brain bank that there is a relationship of any kind? And what was this used in diabetes?
Yeah, so exenatide. So this is a great question, and it's I think one of the things that I think you have a great research question there. That it's sometimes difficult for us to understand these things because like if we're developing a brain bank, we would develop it with patients that have neurodegenerative diseases. If you have diabetes, you're unlikely to be kind of recruited to that type of to be asked to donate your brain. So I think for some of these things, we don't know as well what exactly the relationship is between these, but there is certainly some relationship between having diabetes and having neurodegenerative disease. It's not quite so specific as one-to-one to Parkinson's disease. It's also a risk factor for developing Alzheimer's disease, for example. And I don't think it's well known exactly how that happens, if it's kind of a metabolic disturbance that leads to metabolic disturbances in the brain. I think it's a great question. I'm not as familiar with with kind of the diabetes literature and how those two are directly related. Certainly is a risk factor. So exenatide, actually the proposal for how exenatide would work in Parkinson's disease is different than modulating kind of diabetes related function. It's thought to act through some of these glia that become inflamed and in disease. So most of these are not directly through modulating the disease that they were originally proposed for. It's that they have some secondary effect that may be useful for Parkinson's disease.
I think David has a question.
Yep, we got a question from Zoom. You mentioned that Lewy bodies are a result of a single misfolded protein. Is anything known about the genetics of that protein? Are there any studies connecting it to Parkinson's?
Yes, so I didn't mention that, but and for Parkinson's disease, for example, alpha-synuclein is the misfolded protein. There are also rare familial cases that either have a mutation in alpha-synuclein gene that makes it more likely to misfold, or actually duplications or triplications. So some individuals would just have a higher number of that gene copy, and that is associated also with Parkinson's disease. And those cases, actually, those often lead to early onset Parkinson's disease. So those would kind of accelerate the disease course due to the fact, most likely, that they have a much higher abundance of that misfolded protein.
I've got a sister that's got Parkinson's, and when early onset, it was not diagnosed by her doctor. So how common is it for the general physician practices around to know that somebody may be predisposed to Parkinson's, and how is it diagnosed?
Yeah, so typically, I guess it would depend on your primary care physician, how well versed they are in that. Hopefully they would know to refer you to a neurologist if there's an indication that that's a possibility, especially if there was a family history and you had indicated that there's possibly a family history, then that would be a good indicator for them to refer you to a neurologist. But it's going to depend, I think, a lot on your neurologist and kind of what you tell them or what they may see when you go visit.
And second question, what is the difference between Parkinsonism and Parkinson's?
Yes, so Parkinsonism is a series of symptoms, typically the motor symptoms that we associate with Parkinson's disease, but often it doesn't have a lot of the same comorbidities as Parkinson's disease. We think that's likely because it can affect different systems within the brain. For example, in Parkinson's disease, we know that we lose those dopaminergic neurons in the substantia nigra. In Parkinsonism, you can also have a similar effect, but then often in Parkinson's disease, as I said, this will progress, and often you'll have other symptoms, including often progression to dementia, which does not happen typically with Parkinsonism. It's typically more of a direct motor symptom effect and less of kind of these other comorbidities.
Which would, in my opinion, bring it closer to the familiar or essential tremor. And you got like 3 different progressions, it seems.
Yes, so all of these would have different progression, and some of them would be quite sustained. Maybe there's some symptom, but doesn't progress much. Parkinson's disease, on the other hand, progression is a major part of that disease.
Oh, you had a question. Okay.
I wondered if you could share with us what inspired you to become a researcher, and particularly in Parkinson's disease.
Yeah, sure. So in high school, I was very interested in science. I also was very interested in math. And we had a kind of health sciences class where we learned about all the different organs of the brain. We learned about how the heart works, how the liver works, how the kidney works. When we got to the brain, it was not really known. So there's a lot we know about the brain, but it's an incredibly complex organ. There's a lot we don't know. And so I was incredibly fascinated by that and decided to pursue neuroscience when I went to college. And there, and especially in my graduate school degree, I discovered in understanding the brain that there were really this host of diseases called neurodegenerative diseases that have no cure. So none of these have any disease modifying treatments. We have certain ways to modify the symptoms of disease and alleviate the burden that disease causes, but there's no way to modify the symptoms. So that really, I thought it was a great area of research that could potentially make an impact. And then also I had an aunt who recently passed away with Parkinson's disease. My grandfather also recently passed away with Alzheimer's disease. So I have some personal connection. I realized there's definitely an area where research can make a great impact. Mm-hmm.
I have two sisters who have and had Parkinson's. One was diagnosed at 65 and the other in her mid 70s. If you have that genetic predisposition, you mentioned there were ways to find out if you carry those cells.
Yes. So, so there are certain genetic mutations or genetic variants that can predispose individuals to disease. You can find this out yourself. You can go to 23andMe and find that out, or you can go through your neurologist. The thing is that for right now, there's no kind of clinical indication to have that tested, because if you had that, there's nothing currently that we can do for you differently than we would do if we found out later in disease. The one possible difference is that some of these genetic mutations, we have now treatments that are going to clinical trials, but typically those would aim still to treat quite early disease, but they would not likely be indicated prior to any kind of clinical disease onset. So you can find out, but what you do about it, and whether you want to know and things like that, these are all kind of ethical questions. So, but the main thing right now is that it's not indicated for anyone who comes to clinic, because there's not much different in terms of clinical treatment that we can do for those individuals.
I have these little tests for myself that I do. Does a cup of water jiggle when I move it, or how my walking is? And I guess I'm a little, cognizant of the fact that it's there.
Well, that's very nice, actually, because you would be able to go seek out a neurologist or somebody like that because you're cognizant of the fact that that these may be things you want to look out for. So that's good.
Oh, I have a quick question from Zoom.
Mm-hmm.
For some time, medical diagnosis could not confirm Alzheimer's disease and use the term senile dementia. How are tests today able to confirm Alzheimer's before autopsy?
Yeah, so one of the things that I had told you, Alzheimer's disease in terms of research is a little bit ahead of Parkinson's disease research. And so one of the things that Alzheimer's disease has that we don't have in Parkinson's disease research are those biomarkers. And so while you never get a full confirmation of diagnosis, we can actually be quite sure of the Alzheimer's diagnosis. The percentage of clinicians actually being right is often much higher, because you can use cerebrospinal fluid. We can look for biomarkers there and actually get a pretty good idea if you have Alzheimer's. There are also imaging ligands. So what this is a positron emission tomography, where you can actually get an injection in your blood, and then the clinicians can image in your brain, and we can see those plaques and we can see those tangles actually in the brain and of a living person prior to death. And so some of these are used, especially in a research setting, and in defining some of those clinical trials to make sure, because you wouldn't want to do a clinical trial and it turns out half your patients don't have the disease you're trying to treat. So Alzheimer's disease can actually be fairly well diagnosed pre-mortem now through some of these biomarkers.
I just wondered, is MS a totally different process, or is there some similarity between that and Parkinson's?
Yeah,
and
so multiple
Alzheimer's?
sclerosis can also be neurodegenerative, but it affects primarily the glia that wrap around neurons to enhance their function. So you can get neurodegeneration with that, but the difference is that it's autoimmune, and so it's basically your immune system attacking these glia, these cells that are wrapping around neurons. So it's not primarily neurodegenerative because it's not the neurons themselves that are being affected. But some of the similar processes we do think are occurring, including inflammation, for example. So we've learned some things from multiple sclerosis that we can apply to neurodegeneration, but it is a slightly different process.
My father passed away with Parkinson's, and I believe they said he had aspiration. What is it that causes that? Is it the motor system has failed and allowed reflux or something, or what?
Yeah, so for Parkinson's disease, typically it is not so debilitating that patients will die because of the neurodegenerative disease. They'll have many symptoms associated with that, and that may, for example, for my aunt, left her bedridden for quite a while, but then she had a lot of the symptoms from just having been immobile for so long. It's different than, for example, amyotrophic lateral sclerosis or ALS. In that case, many patients, the motor system is so affected and the neurons that control motor systems are so affected that then it can be difficult to, like, eventually patients are unable to breathe or something else essential like that. Parkinson's disease is not typically like that, but yeah, patients can often have other things associated with that.
I think you had said that less than 25% of the Parkinson's disease cases were caused by genetic factors, and that primarily the causal agents are environmental factors. Who are the main suspects on that side?
Yeah. So for Parkinson's disease, one of the, there's several different things. One can be, for example, kind of head injury. So chronic head injury, for example, Muhammad Ali, boxing. So head injury can be a main exposure. This is why now we monitor so closely. And our football players, for example, if they get concussions, there's concussion protocols because that's known to be deleterious. Pesticides, there are certain pesticides which have been fairly convincingly linked to development of Parkinson's disease. The difficulty with all of these is that it's not a kind of causal factor. You can't just say that anyone who has ever been exposed to the specific pesticide will develop Parkinson's disease. Typically, it has a dose effect. So if you kind of treat your crops every once in a while with this pesticide versus if you're actually out there every day using that pesticide, possibly inhaling it because you weren't using a mask or something like that, it's a dose effect. And then even in that case, it's sometimes difficult to prove that's the causal agent because it's often a mixture of genetics and environmental exposure. So for example, maybe you have to have something that predisposed, maybe it gave, for example, your kind of trash clearance process within the brain was impacted by your genetics. But then on top of that, you have this pesticide exposure that's more likely to kill cells. And so those two together interact, and that is what eventually leads to Parkinson's disease. But some of the main culprits are those that I mentioned. Some infectious diseases have also been linked, but those are all fairly small kind of what we call odds ratio, basically increasing your odds of developing disease. So more in the changing from 1% incidence of developing disease to 2% or something like that.
Professor Henderson, are there any preventative steps, exercise, diet, anything that people could do to reduce their likelihood of getting Parkinson's, especially if they have one of those two genetic markers?
Yeah, so there's nothing that I know specifically for the genetic marker related, but for all of us, I think all the things you hear about quite often that could help you can help you. For example, exercise, but one of the interesting things is also educational attainment. So you're all here today learning. This is all helpful. So it's been shown actually that if you have life dedicated to more educational attainment, often that you will have a much later onset. So even if you did end up getting some of these diseases, especially for the dementia components, this can really offset how long it would be until you develop that. So education, exercise, diet is also can potentially influence these, although it's unclear exactly what type of diet is most influential, but certainly obesity can be linked to a higher incidence of disease. So maintaining a healthy lifestyle in general, I think, has all been shown to set off how long it would be until you get disease.
And looking in the future, I know scientists don't like to make reasonable guesstimates, but when you talked about being able to slow or even stop Parkinson's disease, are we, in your estimation, 5 years away from meaningful treatments, or is it more like 25 years? And, you look around the room and some of us would appreciate hearing it hopefully for about closer to 5.
Yeah, so the lab I came from at Penn, they have been working on this for about 40 years. So Alzheimer's and Parkinson's disease, 45 years maybe. And so I got my kind of insight from them that I could tell how enthusiastic the field has changed so much. So I told you some of these major findings are only in the past 10 or 20 years. So for example, alpha-synuclein was discovered as the main component of Lewy bodies about the same time Van Andel Institute was founded. So that gives you some idea that that's how much things have advanced in the past 20 years. So they were very hopeful. You can see also the clinical trials, these take time. Some of these things we had conceptualized and started going into clinical trials maybe 10, 15 years ago that are now in phase 2 or phase 3. So I'm very hopeful, I would say within 10 years, okay. But I think it's going to be complex also, and that it's not going to be kind of a one shot, everyone, no longer Parkinson's disease is a problem. I think, as she had mentioned, if you've seen a patient with Parkinson's disease, you've seen one patient with Parkinson's disease. There may be different varieties of Parkinson's disease, and these may need different clinical treatments. So I'm hopeful, but I think there's a lot we still have to learn.
We got a Zoom question. How many GMOs impact risks of these diseases? And probably also stuff like pesticides and so forth.
Well, so pesticides, I think for sure I mentioned there's some link. There's no link that I'm aware of any of GMO being linked to any neurodegenerative disease.
Has the recent completion of the human genome sequencing, does that give you more hope that there may be new areas of research? Or is that not related at all to this?
No, it's absolutely related, because essentially before we had a whole genome to reference to, we couldn't really understand, we can find rare variants occasionally, but we don't really know which gene they're in. So basically what the genetic research I told you about, what they do is they will take what's called a SNP array, a single nucleotide polymorphism. They can find several of these throughout the genome, but then they have to reference that to the whole genome. So the whole genome is really essential for that. The difference is that, as I told you, we all have different genetics, right? So when they first did the whole genome, it was from 1 or 2 people. Now we have thousands and thousands of these whole genomes. We get a much better idea of the kind of variety. So for example, you and I may have a lot of differences in our genes, but only a few of those may change our susceptibility to disease. It's understanding which of those it is that really requires huge amounts of genetic data, right? So we would need that. That's why we need thousands and thousands of patients to uncover these differences, because they may, our eye color, our hair color, the genes that encode those that are quite different may not have anything to do with our development of disease. And so, but yes, it's definitely important. Now, some of these studies you can see through Michael J. Fox Foundation or other foundations similar, they're collecting huge amounts of data from, so the Progressive Parkinson's Markers Initiative, they collect huge amounts of data from patients, and this allows us to get closer to understanding what the specific disease-causing genetic or non-genetic changes are.
You mentioned that there's many places around the world that study Parkinson's. What is Van Andel doing that's different or unique from what all the other studies and research is doing?
Yeah, so we all, I think for Van Andel Institute, the goal is to do things that other places can't in ways they won't. And so what that means is essentially things like this MIND initiative, where we are really able to tap into using philanthropy or things like, or the community near us to try to understand Parkinson's disease. So for example, Parkinson's disease in Western Michigan may be different in some ways than in different parts of the world. And so by tapping into the local community with that, by using the philanthropy we have to develop resources, so we have some of the best resources in the world for investigating some of these diseases. And then we still do collaborate a lot with other sites. So one of the initiatives I have, for example, collaborates with my colleagues on the East Coast. And so we do research together. Maybe they have a lot of expertise in kind of understanding how neurons work. And I have a lot of expertise in understanding what pathology does to neurons. And then we can work together to understand that. But Van Andel Institute has done a really great job, in my opinion, recruiting some of the top neurodegenerative disease researchers to this area and really giving them the resources they need to do the most impactful research.
I can add a little bit in
too.
that we're small, and that allows us to be nimble and flexible. And so when there's opportunities for researchers to do newer high-risk areas, we have the ability to move quickly with that. The other thing that I've noticed is because we are small and focused, and our teams of researchers are able to collaborate. So for instance, we have a metabolism and nutrition area, and they're looking at how does that impact neurodegenerative diseases or cancer. And so we're able to integrate some of that research across the Institute. And I think that makes us different and unique. And also our resources allow us to do some different things that other institutes or institutions of higher learning can't do.
So what are some of your successes?
Do you want to speak to that? Or I mean, I can share a little bit about the connection between the appendix and neurodegeneration. And the other one is we are looking at the ability to test pancreatic cancer much earlier than they ever were able to do before. So those are two that come to mind.
Well, in the lean clinical trials that I mentioned, I think that's incredibly impactful. And this has been a major focus both for cancer. There's a similar program in cancer as well to try and like take our best ideas. We can do, think of the best ideas from our models and understanding research. But if we can't bring them to the clinic, we're stuck there. So I think that's a major big impact that the Institute has made is through facilitating these clinical trials.
The pattern for Parkinson's, is there any nationality or that is more prevalent in, or it's one that really doesn't have much or anything like that?
Yeah, so Parkinson's disease occurs across the world. It is definitely different. So different genetic background, for example, which you would expect in different populations across the world, can influence the risk of developing disease, even from some of these environmental factors, or even some of these genetic variants that in some populations, for example, the LARC2 one I mentioned, in certain populations, almost 90% of people with that mutation would get disease. Whereas in other populations, that exact same variant can lead to only 30% of that population getting disease. So for certain, there's different genetic background. There may also be different environmental exposures in different areas, and in different diet and things like that. So a lot of these, so definitely Parkinson's disease incidence changes in different areas, but it is definitely something that occurs in all populations. So it's not as if there's some population that's immune, then we would definitely investigate why they're immune to that, but that's not the case. But definitely it changes depending on genetic, environmental background and things like that.
I have one additional question, and that is, what percentage approximately, Dr. Henderson, of Parkinson's cases lead to dementia?
So approximately 80% of people who have Parkinson's disease sometime before they die will develop dementia.
Thank you. Any additional questions? Well, thank you so much. I don't know who had the record for answering most questions before today, but I know who owns it now. Dr. Henderson, thanks so much. And
Thank you.
And not just for your presentation, but for answering questions for almost 40 minutes. That was superb.